right. Good morning, everybody. Thank you so much for coming to this session. I'm very pleased to welcome Dr. R.T. Mistry, a postdoctoral research associate at King's College London. So welcome to Pittsburgh, R.T. Dr. Mistry's research focuses on the role of the RYR-1 receptor in muscle function during pregnancy, and she's currently a contributing member of a multi-center study on the prevalence of RYR-1-related diseases in the UK and the Netherlands. So we are very excited about that research. Today, she will discuss bleeding issues and abnormalities related to menstruation and pregnancy in the context of RYR-1-RD. So welcome. Thank you, Artie, for being here with us.
Thank you very much, Pat, for your kind introduction. And sorry for trying to start without you.
Oh, that's fine.
Okay, thank you, and good morning. So as you know, I'm R.T. Mistry, and I'm a postdoctoral research associate at King's. And I'm going to be talking to you about my recently published paper on bleeding abnormalities in women with RYR-1-related disorders. So some of you might have already filled out a questionnaire that got distributed by the RYR-1 Foundation about a few years ago now. So these are the results that I want to give back to you, to tell you what we found and what we want to do going forward. Okay, so this is the paper that we'll be discussing today. If you want to get a copy of it, you're very welcome to scan the QR code. Don't worry, if you don't, I won't feel bad. It'll take you straight to the journal website, okay? So about 30 years ago, the RYR-1-related disorders mainly involved central core disease, and it was really considered to be rare. However, looking at the landscape of RYR-1-related disorders, we now have a more wider clinical spectrum. We have the very much clinically relevant congenital myopathy-like presentations, but also an expanding range of malignant hypothermia-related presentations, of which exertional rhabdomyolysis and myalgia are the most common. And in addition, we're now seeing non-neuromuscular manifestations such as a bleeding phenotype. So this slide illustrates the role of the ryanodine receptor in muscle. The key thing to know here is that the role is to release calcium from the inner cell machinery to the outer cell space, which then triggers cells to contract and therefore muscle contraction. So you can see, like, when a mutation in the RYR1 gene occurs, the protein dysfunction can cause quite a lot of damage. In fact, you might hear that mutations in RYR1 and disorders related to RYR1 are heterogeneous. This is because different mutations can cause different protein dysfunction and therefore a different clinical outcome. This is also apparent when mutations are in dominant form, so that's one mutation causing the disorder, or in recessive form, so that's two mutations required to cause a disorder. So about 10 years ago, in both mice and humans, and in particular females, it was recognized that there was a bleeding phenotype associated with RYR1. Although central core disease had been known for 30 years, this phenotype had not been previously reported. Because RYR1-related disorders are typically neuromuscular in condition, people tend to focus on the neuromuscular effects, but other symptoms may be getting lost. Some of you know Professor Heinz Jungbluth. He's the one who, in fact, brought this to light with the bleeding phenotype that he observed in one of the large families that he works with, in which all of the females would describe a bleeding phenomenon. So previously, the mild bleeding disorder has been described in a small cohort of RYR1 MH mutation-carrying patients. The bleeding was characterized by excessively heavy periods and severe postpartum bleeding, and so that's bleeding after childbirth. This was initially anecdotally found in a few selected patients, and then that's what prompted further research. So the group in 2000 2016, they used a tool called the Molecular and Clinical Markers for Diagnosis and Management of Type 1 von Willebrand disease. The questionnaire has a really long name and is almost always abbreviated, so I'm only going to refer to it as the MCMDM von Willebrand disease questionnaire. This questionnaire is a questionnaire that's used by doctors to determine how easily you bleed and then to gain insight into whether or not there may be abnormal bleeding. It asks about bleeding tendencies. For example, an increased tendency to bleed after surgery would then increase your score, and vice versa. In this study, they found that people with RYR1 mutations were more likely to get a score above 4, which would then be pathogenic, meaning that they are likely to have a bleeding disorder. The authors then looked to confirm the findings in a malignant hypothermia mutation carrying mouse model. To determine if there was indeed a bleeding phenotype in this mouse model, they used tail artery bleeding times. So for this, a small cut was made in the tail artery, and the time to the end of bleeding was then recorded. Don't worry, it's not particularly harmful. Like, this was passed by the government to do this research, so don't worry. The animals are fine. You might notice that there's a similar pattern to the bleeding questionnaire findings. In the Y522S mouse, I don't know if my... So the Y522S mouse here, so these are the ones with the mutation, so that's the malignant hypothermia mutation. They had a slightly longer bleeding time than the non-mutated mice. Then in another set of experiments here, the animals were pre-treated with dantrolene, which is a specific agonist that reduces the calcium release from the ryanodine receptor channel. And this is particularly important when the RY1 mutation causes increased calcium release, causing problems in the body. And when they were pre-treated with dantrolene, the bleeding time was reverted almost back to normal. So you can see here, it's basically like the wild type being normal. And that's evident when you also pour the vehicle control. So that's the same mixture, just without dantrolene. We see almost the same impact as no dantrolene. Does that make sense? So how's this possible? You might be asking. It's important to understand that the RYR1 gene is widely expressed in the body, even in small numbers. We know it as a skeletal muscle calcium channel, and it's most widely studied in the skeletal muscle. But we also know that it's expressed in many other tissues, from the artery, brain, to prostate, to the vagina. It's important because mutations in the RY1 proteins that we see may cause problems in other tissues that they're being expressed in, so not just in skeletal muscle. Okay. We've had to change the way we present this because the videos were not working in the original presentation. So here we've got some calcium imaging videos where the calcium molecule has been tagged with a green fluorescent dye, and that helps us visualize its movement within a live muscle cell. So looking at the function of a normal ryanodine receptor channel in the smooth muscle that lines the blood vessels, we can see the sparks of calcium release from the wild-type non-mutated RYR1 channels. So this is how frequently you would normally see this calcium release, right? In the Y522S mutated smooth muscle from the arteries, we can see that the sparks are a lot more frequent. Can you see that? Yeah? Yeah, you just see those sparks a little bit more often. And then we can see in the final video, when the same cell, so smooth muscle from the artery, is then pre-treated with dantrolene, those sparks are almost diminished, really. I've played it a few times just because it's quite a short video. Yeah. And that means that this bleeding abnormality may be reversible with dantrolene in the MH-susceptible Y522S mouse model. Forgive me, I'm going to have to go through the slides again. So briefly, we'll talk about the possible mechanism of action. Upon injury to the artery, the smooth muscle in the artery would normally contract and in an attempt to reduce bleeding. However, RYR1 acts in the opposite way in smooth muscles than it does in skeletal muscle. So in the skeletal muscle, it increases contractility, but in the smooth muscle, it reduces contractions. Therefore, in the case of the artery, which is lined with this smooth muscle, specific mutations in RYR1 can cause increased relaxation and therefore increased bleeding. This is the theory that we're working with right now. So bringing back the focus to humans and women's health, heavy menstrual bleeding is currently clinically defined as the menstrual blood loss that interferes with a woman's physical, emotional, social, and material quality of life. Annually, 5 to 24% of the female population will consult their GP for heavy menstrual bleeding, and causes are found in about 5% that seek medical treatment, but almost 50% are left without an explanation. So what did we do? We designed a 15-minute online questionnaire which women could answer themselves. It was based on the validated bleeding assessment tool, so the MCM-DM von Willebrand disease questionnaire, and the NHS heavy bleeding assessment tool that was then modified and rewritten in non-medical language. So the questionnaire didn't need to be administered by a clinician, and people were able to answer it themselves. The questions were then expanded to include obstetric history, fertility, gestation, postnatal bleeding, and even baby birth weight. The questionnaire was then trialed in preterm birth patient participation groups, and they informed us if there were any questions that needed to be replaced in terms of sensitivity, comprehension, and even advice on the structure and length of the questionnaire. So after being granted ethical approval by the University Ethics Board, we were then able to begin the study. We recruited patients via patient organizations like the RYR-1 Foundation, through treating doctors, social media, and the university, the latter being more for the controls. In the end, we received 154 responses, and we analyzed those responses between RYR1 carriers and control groups, and we set a significance threshold of 0.05, which is the commonly accepted p-value in scientific studies. So to give you a summary of what our participants looked like, of the 154 participants, we had 66 RYR1 variant carriers and 88 controls, and some of these participants are in this room today, so it's lovely to meet you. To give you a little bit more detail about these participants who answered the questionnaire, of those who had RYR1 diagnoses, 35 people knew that they had a dominant mutation, so one mutation causing their disorder, and then 13 people had recessive mutations, so that's 2 mutations causing their disorder. The most common diagnoses were central core disease and malignant hypothermia susceptibility, which is within the expected range. We believe that the results of this study are genuinely due to the disease-causing mutations, but there are certain differences between the groups that we should take seriously and just take into consideration when interpreting the results. So, for example, we have the age of the control population, which is slightly skewed to a younger cohort. This is important to factor that a younger population may not have had many pregnancies, and an older population is likely to have more medical conditions. Another example of that is the ethnicities within the groups. Both were distributed differently, and then this could reflect the different genetic predisposition to disorders. But after analysing these results in detail, I don't believe that these factors impacted the results, and therefore the differences are due to RYR1. But this is being shown to you for transparency. So, using the modified bleeding questionnaire, we see a similar trend to the results that we see in the previous paper by Lopez et al. We find that women with mutations in RYR1 had a higher likelihood of having a pathogenic bleeding score, so a score above 4. This was in 48% of the RYR1 group, whereas only 12% of the RYR1 group had a pathogenic score, meaning that these participants were likely to have a bleeding disorder. Even further, we had a look at the recessive and dominant mutations, and we found that there was no statistical difference between the bleeding scores. So it's important to note that this questionnaire relies on the correct reporting of the genotype by patients. So we may see a trend. As you can see, the dominant mutation carrying patients had a slightly higher score. So we see that trend, and there's a chance that that trend could be more pronounced if the genotypes were verified by a clinician. So using several questions from the NHS bleeding assessment tool, heavy bleeding assessment tool, we had questions on different behavior changes during menstruation, so periods. We found that participants with RYR1 mutations were more likely to avoid traveling and to change sanitary products overnight. That really indicates a much heavier bleeding phenomenon in these patients. So this is some data that was not actually included in the published paper. It's actually from my thesis, where I looked at the contractility of the uterus from non-pregnant wild type, so that's non-mutated, and mutated Y522S mice. So this is the same mice that was studied in the other paper. So the uterus strips were taken, extracted from the mouse, and tied to an instrument that detects any contractions and then converts that to tension data. So, for example, you'll see each time there's a contraction, there's a spike here. And this is normal. Your uterus is always contracting. You might not feel it, but it's always contracting, even when the animal is not pregnant and not in labor. Same for humans. And so we found that the heterozygous tissues had more frequent and shorter contractions compared to the wild-type tissues. So that's the two differences here. So increased frequency of contractions and then shorter contraction duration. And you can see that here. The peaks are much shorter and then also closer together. So we believe that in this finding could in part account for the heavy menstrual bleeding in the MH mutation carriers. Basically, if you're not pregnant and your uterus is contracting more, it's going to be expelling the uterine lining more often. In this questionnaire, we also wanted to know if having an RYR1 mutation could increase the risk of having pregnancy complications. The most common problems in the RYR1 group were placenta previa, so that's when the placenta is covering the cervix or it's a low-lying placenta. So this can lead to excessive bleeding. We also saw that these patients were more likely to have preeclampsia, so that's the high blood pressure that develops during pregnancy or postpartum and is linked to various other medical conditions that can be harmful to mum and baby. We also investigated birth weight in this paper. One of the reasons why we included this is because animal research that I was conducting at the time was suggestive of the heterozygous, so the mutation-carrying mice, giving birth to lower weight pups. From this questionnaire, we then found that women with RYR1 mutations would give birth to lower weight babies compared to the control group. supporting the notion that RY1 mutations could be leading to restricted fetal development. However, we can't be 100% sure if it's due to the shorter pregnancy or the mutation. I'll come to this in the next slide. In a separate cohort of MH patients from the Netherlands, we found that babies that had RY1 mutations had a lower birth weight than those that did not have mutations. And this cohort was actually matched for gestational age and therefore maybe a more accurate representation of the changes in birth weight. So we also hypothesized that if RY1 mutations impact the uterine contractility, this may impact the onset and the length of labor. Through this questionnaire, we did find that pregnancies were generally shorter in the RY1 group compared to the control group, indicating that the RY1 mutation might play a role in the initiation of labor. But I believe, personally, that a lot more research needs to go in this with a more refined approach. Like I said, this is preliminary research, and this can be done to a much greater in-depth extent. The RY1 participants were also more likely to have a planned C-section. Although it should be noted that women with RY1 mutations or those that have had multiple children may already be candidates for C-sections. In this study, although not statistically significant, because we had very few responses to this question, there was a history of more severe and more frequent postpartum bleeding in the RY1 group. In the RY1 mutated cohort, the reported blood loss volumes by 3 participants are clinically categorized as major to severe postpartum hemorrhage, compared to only 2 cases of minor postpartum hemorrhage in the control group and the RY1 mutated cohort. In this population, in the general population, spontaneous preterm, I was going to say preterm birth because that's what I'm used. Spontaneous postpartum hemorrhage is estimated to occur in about 5% of the population. So this indicates an increased risk of severe postpartum hemorrhage compared to the in the RYR1 group compared to the control group, which is representative of the general population. And we can really see that here. Like, if we look at the incidence of postpartum hemorrhage, we've got about something representing the general population in the control cohort. I do believe that if we had more N, we would have seen a statistical difference between these 2 groups. The RY1 mutated patients were also more likely to report bowel and bladder problems, with common symptoms including constipation, diarrhea, and urinary incontinence, potential indicators of dysfunctional colonic and urinary bladder smooth muscle function. So as we know now, the recessive and dominant mutations can cause different clinical presentations. So we also wanted to know if this was apparent in the suspected bleeding disorder. We found that individuals with the autosomal dominant mutations were more likely to have bleeding between periods and babies with lower birth weights compared to the participants with autosomal recessive mutations. In line with previously found results, this study does emphasize that the RYR1 mutations that are autosomal dominant and are gain of function are more likely to be frequently associated with excessive bleeding and other gynecological problems. So that's similar to the Lopez paper mentioned before. So this study clearly demonstrates that the pathogenic RY1 mutations are associated with prolonged bleeding, obstetric, gynecological, and bowel and bladder symptoms. We believe that RYR1 mutated patients are likely to have a bleeding disorder that manifests as heavy menstrual bleeding, postpartum hemorrhage, preterm birth, and low birth weight, in addition to the GI tract dysfunction. Considering that most of the individuals carrying MH-associated variants are largely asymptomatic unless they're exposed to general anesthesia, it's conceivable that there may be many more females and males with an undiagnosed bleeding disorder due to this genetic background, predisposing them to this range of complications. We were also able to establish a tentative genotype-phenotype correlation for the RYR-1-related bleeding disorder. I believe that the work's not finished and there's still a lot more to do. So to really demonstrate to the scientific, clinical, and wider community that there's a bleeding phenotype associated with RYR1 mutations, I believe the first thing to do is to conduct a prospective study. So that's to collect information from current patients with verified RYR1 genotypes and their ongoing experiences with bleeding. Considering the improvement of the bleeding phenotype following dantrolene treatment in the MH animal model, a clinical study assessing the potential improvement of heavy menstrual bleeding symptoms in patients with unexplained heavy menstrual bleeding should be considered in the future. We should also look to investigate the uterine contractility in animal models and in the non-pregnant human tissues, for example, those collected from hysterectomies or C-sections, because I think it would be good to further characterize because I think it would be very good to understand why we are seeing these differences in the bleeding phenotype. I think we should also characterize the gastrointestinal problems and the genotype correlations going forward. So thank you everyone for listening, and a very special thank you to all the participants and the collaborators on the study, and a very big thank you to the RY1 Foundation for supporting this study. I'm happy to take any questions. If you don't feel comfortable asking me now, ask me later. I've put my email address up there, so please get in contact. I don't mind. Just to preface, I'm not a clinician. So if you do have more clinical questions, I will put you in the direction of Professor Heinz Jungluth, who really put all this work forward. So thank you. If you have a question, we have a mic. So if there's anybody.
So you talked a lot about complications during the pregnancy. Did you see a higher prevalence of miscarriages in the RYR1 variant patients, or?
Yeah, so that's a really good question. We actually did ask about miscarriages, and we really broke it down to miscarriages before 16 weeks, because we have a lot more miscarriages before 16 weeks than people know of, but also later on in pregnancy. Unfortunately, we didn't see any difference. I do believe that this may be due to the low participant numbers. From my memory right now, when I was analyzing this data, I remembered there weren't many responses regarding miscarriage. We also, before that question was on the questionnaire, we also asked participants if they would like to answer that question, because we understand it's a very sensitive topic, and some people chose not to. So I think if we had a larger sample size, we may have more data, but I can't actually comment on the incidence of miscarriage. Yeah. But scientifically, we did have some basis to study it.
I just have a quick question. Have you guys looked into asking them about birth control with this because like I've been taking it because I had really bad like excessive bleeding and terrible cramps, and it was brought down from like 8 days of just excessive bleeding to only 4 or like 5. And I think it was a pretty good treatment option for me, so I don't know if that would work for other people.
Yeah, and I think commonly in the clinical setting, birth control is prescribed as a way to control for heavy bleeding, and I think that's going to continue regardless of your genetic predisposition. In the study, we did ask about birth control because we wanted to make sure we wanted to make sure that there were no other confounding variables. So if a patient was having extremely heavy postpartum hemorrhaging, but they didn't have very heavy periods, we would want to know why, and that birth control question would then tell us. Yeah. In terms of the number of participants being on birth control and not, we didn't see much difference in those responses.
So to kind of go off of Courtney, I have an 11-year-old that has RYR1. Would you suggest talking to their physician before they start their cycle to maybe get them on birth control to see, like, maybe help it before it starts? Or would you recommend waiting until she actually starts and see how or what? Or
Yeah, I think, so first of all, being a PhD and not an MD, I can't give you clinical advice. I can just tell you what we've found. I personally would inform your physician that this is something that we've seen. There are studies on it. You can take them my paper. Tell them that we understand that women with MH-associated, so dominant mutations, could be more likely to have heavier periods. But pre-treating with birth control is something that I can't advise on. Thank you. Yeah, do you want to just take the mic?
I know there were a few recessive people in your study, or carriers in your study. Do you plan to look more closely at the recessive mutations to see if there is a difference and how their bleeding is? I know my daughter recently started her period, and initially they were really heavy.
Yeah.
But not every single one. But I was actually really surprised, especially with the first 2, how heavy they were, because it was like full-on periods for her first one, which I was not expecting.
And
so,
we normally see the opposite, in fact, don't we?
Yeah, right. Yeah. So, but it was full-on. So I was wondering. Now her last one was really light, but I mean, she's only been on it for like 6 months. So are you going to continue to study like the recessive mutations to see? But also, is there some sort of a tracking? Like I think she uses an app on her phone to kind of see the length. But would there be something that would be better to track like the heaviness, just so we can kind of be prepared and know what to look for, so we can?
Yeah.
Stardust is a great app to track.
What is?
It's called Stardust. I use
Okay.
it. And if you like pay for like a premium subscription, it'll give you like the food that she wants to eat, the heaviness that you should expect for mood, and like everything. If you do like partner mode.
She is moody. It sure is.
You can like share like how you are with your friends through like partner mode. So like me and my best friend have it set up together.
Yeah. Can I ask which app she's using?
I don't know. She might just be using what's on her phone.
Okay. Because the one that I know is most common is Flow. You might have heard of Flow. But if I can't, I don't use one myself, so I'm not that well versed into using it. But pen and paper. If all else fails, pen and paper. In response to your question regarding the genotype differences, so that's why we believe the first thing to do is a prospective study. So clinicians like Hines and anyone that's seeing someone in the clinic can start collecting this data now going forward. So we have a genetic verification, so exactly what they have, and then we can get more detailed menstrual, but also say the patient then grows up to have an adult neurologist, decides they want to get pregnant, we can then follow through. That would be really, really useful. So that's the first thing we should do. I think it would be really useful, but we need funding to do that. So that's my job now, is to apply for funding. But that's the aim, because, like I said, although so far the animal studies have been done in a dominant RYR1 MH mutation mouse model, there's no reason to suggest that there may be some bleeding phenotypes in the RYR1 recessive mouse model or humans. It's just about teasing out that difference and really understanding how it affects the protein and how that might affect the contractions in the uterus or for example, other hormones in the uterus, such as oxytocin, estrogen. That will, yeah. Hi. Can I? So the question from Avi is, how would this be screen timeout. Sorry. How would this be affected by going on T? Would it be affected? I'm not sure what T is. I'm so sorry. On testosterone? Okay. At the moment, I don't know. I'm very sorry. That's the most honest answer that I can give you, because I personally don't know how testosterone interacts with the RYR1 protein. And I'm not sure if there may be some studies out there. Yeah? Yeah, please pick up the mic.
I looked into that because I have a friend who's transgender, and it did kind of go away for him. And also, I read some papers, and I found a couple that said that it kind of sterilizes you in a way and changes how your body works. Mm-hmm. So I thought that was interesting. But that was just one study, so I haven't looked into it too much, so I'm not really sure. But I thought that was interesting.
I had my son with a natural birth, with induction, and the day after I have an episode of rhabdo. And even if the labor was very short, because it was only 2-hour labor, so a short labor, have you thought of studying the correlation of the muscular effort during the labor and the possibility of having rhabdo? Do you think it could be a future study to study this correlation?
Can I ask you to repeat that, please?
Yeah, it's the question is about the correlation of the muscular effort during the labor and the possibility to have rhabdo episodes the day after or days after.
No, that's not something that I've considered. Not in relation to this study at all. No.
Okay, it's only a question. Thank you.
No, it's okay. Sorry.
Hi. So first question, I just want to make sure I'm clear. Are you saying that the reason why the periods can be heavier is just because of the increased contractions?
So that is one of the reasons that one of the findings from the mouse model indicated increased frequency and shorter duration of contractions. Now, because we haven't studied that in humans, if we could study the contractility in humans, that would be more of an indication as to why this is just one of the possible mechanisms that could be causing, because we know that increased contractility of the uterus can cause heavier bleeding, but there are other mechanisms that can cause increased bleeding. Does that make sense?
Yeah. And my second question was about pregnancy. So how did you guys determine what conditions that you were going to ask about? So you mentioned like previa and other things, and because like one that you didn't mention that is really heavy bleeding after for different reasons, I guess, is like accreta, percreta, where it attaches. And so I'm just curious how many RYR1 patients or with related disorders would say that they had an episode, something like that, or hysterectomy.
Yeah.
So just curious if you're going to do more, and if so, would you consider like adding things like that?
Yeah. So I know that hysterectomies have been unfortunately quite frequent in women that have had really heavy bleeding. This is anecdotal evidence from clinicians. We did ask about hysterectomies in the study, but we've not found anything in this study. Sorry, I forget your other part.
The other one was like accreta. It's 3 different levels, but where it attaches and, it causes crazy amounts of bleeding.
Yeah, so placenta previa can be associated with placenta accreta. We didn't ask about accreta specifically, and I believe it's because the midwife that was advising on this project, she said these are probably the most common ones that you want to be asking for, but there's no reason why we can't ask about that in the future. Yeah.
I'm sorry. One question. I just was curious. Have there been any reports of extended bleeding when traveling? Like, for some reason, I noticed when I was flying, my period was a lot more heavier, and I got through pads and tampons more than I normally would. Is that a thing, or is that just me?
So was your question, is there a reason why your periods may be heavier when traveling?
Yes, like flying, is that, I don't know, sometimes altitude, has that been an issue for other people?
So in relation to this study, we asked about whether they would avoid traveling as one of the behaviors, because you would potentially feel uncomfortable traveling because of the heaviness of your period. We didn't ask about how heavy the periods were when traveling. So I can't give you an answer in relation to RY1 diseases specifically, because that's not something that we studied, unfortunately. But I can understand that heavier bleeding during traveling could be due to the increase in exercise.
Okay.
Yeah.
And one more thing, as I get older, I feel that they get worse.
Sorry, could you repeat that, please?
As I get older, it feels like my periods get worse.
Oh, as you've got older, your periods have got worse, is that?
Yes.
Yeah, the same thing happened to me.
Yeah. Yeah, again, it's not something that we've looked at over time.
Okay.
So, yeah, I'm not sure why that would be happening over time.
Okay.
Yeah. Okay. No problem. Have a chat with me if you want later. Sorry if I couldn't answer all your questions, but I hope you did get something out of this talk. Thank you.